My lab is interested in the cellular pathogenesis of lower urinary tract disease. So when I started my faculty position 7 years ago, there was really not much available in terms of human tissue to do translational research, especially intact human tissue. And so I started looking for partners to collect lower urinary tracts from normal men and from men with disease. And so we've been basically taking anatomical portion by anatomical portion and doing single cell RNA sequencing to characterize the different cell types that are in each of these different anatomical areas in both normal and diseased, and trying to understand changes in each of the cell types. So this all started with our interest in BPH. We know that it's anatomically restricted to the transition zone, as you can see in this 49-year-old man here. And so we wanted to know whether or not there were different cells in there. And so what I'm going to show you is just a couple of things that we've published over the last few years that have large human data sets associated with them. I'm a member of the GoodMap Consortium, and so they have a database there. You'll see these links throughout the talk. What you can do is click on these links as they're available. I'll also show you on our website where these links are available, and they will take you to a whole collection of the data that you can easily browse or download. It will contain FASTQ, raw data files. You can also get SORAD objects where we've annotated our single cell data from that. It'll show you fax data and different antibodies we use to build these panels that sort out specific cell types, et cetera. And so this just gives you a view of the tissues that we are collecting. So these are from a local transplant organization. We have a lower urinary tract from an 18-year-old man shown here. We dissected into these different anatomical portions, and then you can see a transverse section that's stained as a whole mount of a human prostate. And so we have sagittal sections, transverse sections of all of these different organ types for those that are interested. Okay, the next study we did, we actually expanded a little bit on the cell types. We published this just this last year, and we were able to show that there are subtypes of fibroblasts and smooth muscle. So if you subcluster these, we can get a little bit more detail on the subtypes of fibroblast and muscle, and we could show their differential distribution. So if you go to our website, you'll actually be able to see from this particular study better annotation of these subclusters, and you can browse the single cell data from both the normal and look at the same cell types in benign prostatic hyperplasia. In addition to that, we have annotated all of the immune populations so that these can actually be used as a reference for any studies where you produce your own single cell data. In addition to that, we have lists of receptor ligand interactions between fibroblasts and immune cells. You can see the strength of those interactions so that you can come up with hypotheses about how these cells interact, which is really nice when you take an objective look through single cell RNA sequencing data. We've also created stromal cell atlases of the mouse prostate and urethra and compared them to their human counterparts. You can download those data and browse those data as well. We've also created atlases of the mouse prostate and urethra and looked at epithelial cell types. We found a couple of new epithelial cell types. Again, we compared these cell types to their human counterparts, and you can find differentially expressed genes, et cetera, at this link, and you can also browse the single cell data at our website. We also have a collection of over 700 human BPH specimens and 150 of these normal adult specimens so that you can start doing immunohistochemistry studies on markers that you're interested in. So this kind of gives you a look at what we do here, and what we've collected, we have, like I said, 150 male lower urinary tract specimens from organ donors. On the right-hand side, you can kind of see the age and ethnicity distribution of our organ donor repository. So you can do studies where you look at the spectrum of aging and disease. We have large and small OCT blocks of these specimens. We have large and small paraffin blocks. You can see a transverse human bladder in this big block. You can see a sagittal human lower urinary tract. So this is the bladder, this is the prostate, and then we have small and large BPH blocks. We also have blocks from obstructed bladder specimens. So men who are undergoing simple prostatectomy for BPH, we also take a section of bladder. We have clinical annotation of these, so you'll know whether or not they have detrusor overactivity or underactivity. And then we've also produced a cellular atlas of the normal adult male bladder and compared it to the mouse. We have markers of each of these. These data are not yet available, but they will be soon. And then we've recently started collecting female lower urinary tract as well, and we're creating a male-female comparison of the bladder and urethra. We also have produced a ureter atlas. These things are in process and will become available eventually. And then finally, I just wanted to mention that we are doing spatial transcriptomics in the human lower urinary tract using these specimens that I just showed you. This was a Convergence Award that we received from Caribou. And we've successfully done this in human prostate. We have a paper that should be published by the end of the year where we use this nanostring platform to segment specific cell types that we were interested in human prostate. And you can see here that we're also doing this in human bladder. So each of these regions of interest, you can segment specific cell types out and capture the RNA from those. And these are in FFPE blocks. If anybody is interested in a collaboration here and you have a specific hypothesis that you want to test, we're happy to help you do that using our specimens and generating the data for you here at UT Southwestern. All right, so with that, I wanted to say thank you. I don't know how much time I have left, but I'm good. Okay, so the last thing I wanted to do actually is show you our website. All right, do you see that? Do you see our website up there? Okay, so on the left-hand side, you can see we have tabs for publications, biorepository, and single cell data. So I'll just kind of go through these. Can you see the publications tab now? Okay, so if you look at each of these publications, you can see that there's a link for the GoodMap repository for all of the data associated with these publications. And then there are GEO links for the raw data that if you know bioinformatics, you can manage this yourself. If you want SORAT objects, we can send you those as well. And then if you click on single cell data, you can actually open up different data sets. So here's an example of all of the different cell types from our recent Journal of Pathology paper on BPH. And you can see all the different clusters are annotated. And then if you put in a gene like KLK3, which is PSA, you can see that it's highest expressed in the luminal epithelia. So if you want to browse a gene of interest, this is a good place to do that. And then finally, the biorepository. So like I said, we have over 700 patients that you can look through. Here you can filter these by disease phenotype or clinical features. So here I've selected only BPH patients that have diabetes and were on a 5-alpha reductase inhibitor. And then we have actually scanned the H&Es from each of these patients and created a little thumbnail so you can at least see the histology associated with this. If you want higher resolution images, we can send those to you as well. And so we have disease, the ID, age, ethnicity, So if you want to start looking at differences in ethnicity or age or medication, diabetes, et cetera, then we have a large enough repository now that you should be able to statistically study the question of interest. So with that, I'll say thank you to the people in my group. Dia is a postdoc in the group that's just taking a faculty position at the beginning of next year at InStem. Gervais Henry has left us and is now as at DNA Nexus. I wanted to thank Kathy's lab at Columbia, and then especially the Southwest Transplant Alliance, where we collect these lower urinary tracts from males and females now, and then of course funding from the NIDDK. Thank you very much.